Good afternoon, everyone, and welcome to our distinguished lecture. I'm Frank LaFurla. I'm the Dean of the School of Biological Sciences here, and we have a really fascinating talk today on the microbiome, and I can tell you that today's event is actually a family event because in addition to Jennifer's husband, her kids are here, and her mother's here, and even her sister-in-law's here from Denmark. So welcome, everyone. I think you're going to be very proud of Professor Martiny. So let me just always, as I'd like to begin these things, acknowledge the wonderful staff in the Dean's office for all the hard work in putting these events together. Why don't we give them a round of applause, because without them, this would never be able to happen. So as many of you know, our foundational theme in the School of Biological Sciences is focused on mind, body, world. The concept is pretty simple here. And that is that life is interconnected, and that for many of us, to have a healthy mind means we need to have a healthy body. And to have a healthy body, we need to have a healthy world. And today's talk will be focused on actually probably all three aspects, but we like to partition it into the world aspect. And not all elements of our world are visible. In fact, perhaps some of the most important aspects of our world are invisible to the human eye. And it's in that spirit that Professor Martiny is going to talk about the microbial world and its impact on our health and the health of the environment. So in addition to being a professor in the Department of Ecology and Evolutionary Biology, she is also the director of the newly established UCI Microbiome Initiative. She received her bachelor's degree from UC San Diego, and then went on to receive her PhD at Stanford before going on to Oregon to do a postdoctoral fellowship, eventually landing at Brown University, where she started as a faculty member, and then came here to UC Irvine in 2006, and she has just had a really meteoric career since then. Over the years, she's gotten many honors for her work, including a career award, very prestigious career award from the National Science Foundation. She was a Gordon and Betty Moore Foundation Junior Investigator, and most recently was named as a Fellow of the Ecological Society of America and of the American Society for Microbiology. As the director of the Microbiome Initiative, her research focuses on the generation and maintenance of microbial diversity, which involves studying all microorganisms, viruses, bacteria, and fungi. And what's really cool about her area of research is that she doesn't distinguish between the human body and the environment, and neither do the microbes. So with that, let's welcome Professor Martiny.
Well, thank you everybody for coming today. It's such a pleasure and honor to be here to tell you about my research. And I want to start by thanking my family and friends, many of whom are here today, for their support, and also to Frank and the school for this lecture series and inviting me to have the opportunity to speak to you today. You know, I've really enjoyed this lectureship series. I look forward to them every quarter, although I have to say I have mixed feelings about this one, but I'm sure it'll go okay. Anyway, I want to tell you about microbes. As Frank said, they're invisible to the naked eye, so most of us don't feel very familiar with them. Some of the beautiful renditions of them I've put up here, and they include some that you might have heard about before, bacteria and viruses, also fungi. Luckily, some of the fungi, like these mushrooms up in the corner, have macroscopic forms that are very delicious to us, and we enjoy those. And there's many others that you might not have heard of, like diatoms that you find in the ocean and lakes. And today, if you just take two things away from this talk, I hope you take away these two things. First, microbes are really abundant and they're everywhere. But the good thing about them is that only a very small fraction of them actually can infect us and harm us. So, you know, the traditional germs that you think about those are a very minute fraction of all the microbes out there. And secondly, microbes are incredibly diverse. They're diverse in terms of the number of different types that are out there, but also in what they do, their physiology, how they make a living, the environments that they can survive in. In fact, we discover new ones and new ways of making a living all the time. And that's why I think microbiology is one of the most exciting aspects of science. We still are in this discovery stage. So traditional microbiology, however, in the medical side of things, have really thought about microbes one at a time because they're thinking about one pathogen at a time. And so one of the guiding principles of that field has been this idea of Koch's postulates, that in order to figure out whether a microbe is in fact a disease-causing agent, you have to infect an animal, be able to re-isolate the microbe out of that infected animal, inoculate a new animal, see that it comes down with the same symptoms, and then again be able to isolate the organism again. And these principles have served us really well for a very long time. But in contrast to this part of microbiology, environmental microbiology has really focused on communities of organisms. And so just like humans can have communities, microbes have communities too. And lately, the buzzword of these communities is microbiomes. You may have heard about them in the popular press. And all this means is a collection, a community of microbes such as bacteria, fungi, and viruses. And so environmental microbiologists, when they went outside in the environment, not thinking about humans, and isolating different organisms, for instance, in these types of columns here, they could see that there was a tremendous diversity. You can even see the colors here in this culture. And they understood very quickly that many of these microbes couldn't grow on their own. One strain would be eating off of another strain's byproducts, for instance. And so you really couldn't understand a microbe without understanding its community. Now, this picture may look to you like a far-off galaxy, and it's one of my favorite pictures from my lab. It's actually just a drop of seawater stained with DNA stain under a microscope. And what looks like the big planets in this picture are actually just bacteria, and the small faraway stars, those are viruses. So actually in a glass of seawater, if you took a glass of seawater from the surf around here, there are more than 1 billion viruses in that glass of seawater. So when you go swimming next time, you can think about that in the ocean. But again, the lucky thing is that these viruses, they don't infect humans. In fact, they infect these bacteria. And so there's this whole other world out there of all this diversity that we're really still discovering what it does. The other thing that this picture does is it reminds me how difficult it is if you just had a microscope to try to enumerate and characterize the diversity of organisms, microorganisms, right? Because pretty much I can only say there are 2 different types here, and one's larger or smaller than the other. And so it really wasn't until DNA sequencing and the revolution around this new technology where we can sequence quite cheaply and fast the DNA out of any sample, and then we could start characterizing the different microorganisms there. And so that could be soil, hot springs, wastewater sewage plant, and this nice picture, which I wonder if you can guess what it is. It's actually a shower curtain, and the grime on a shower curtain, there's a very nice study that went out and scraped the grime off of people's shower curtains and characterized that diversity. And so environmental microbiologists have really literally had many field days characterizing all the microbes out there just in the last 20 or so years. So ecologists, though, they really like to study the distribution of organisms. And here what I have up is just a plot of the diversity of bird species all over the world. And the darker colors represent higher diversity areas, mostly in the tropics, and the lighter colors, fewer species. And so when I got my PhD, around the turn of the century now, there really wasn't any map like this for microbes, not one. So we pretty much knew nothing about how microbes are distributed across the planet. And so, again, DNA sequencing then opened up this whole area for ecologists to go wild and start to understand how these organisms are distributed. And so I was really excited then when this paper came out in Science that showed that the number of species of bacteria seemed to vary across different habitat types, like forest soil or marine sediment or ponds. And you'll notice, if you're scientists in the audience, you'll notice there are no error bars here, because they literally had one sample from each of these habitats, and they published it in Science. So that's how new this was. But still, some of these trends kind of still hold. And so this is one type of diversity that ecologists are interested in, called richness, just the number of types. But another type of diversity that we really care about is composition. So if we think about bacterial diversity, then we also want to know not just how many are there, but what different types there are. So in these pie charts then, imagine each color is a different species, and the relative abundance in your sample is represented in the pie chart. So what you can see here is, in this mock community, that the two grassland samples, they're not exactly the same composition, but they're very similar, as you might expect, because they come from the same habitat. Whereas the desert community then, it might have different species, and it might have different relative abundance of some of the shared species. All right, so this is pretty easy to wrap your head around, but there's only so many pie charts that you can look at, and often we're dealing with thousands of samples. So we have to plot this kind of information in a different way. And I'm going to walk you through one of these plots, because I'm going to show quite a few of them today. But real quickly, then, if you have a pie chart, and let's say it's a grassland composition, I might place this then on this plot in the corner here. And the second grassland sample, because it's quite similar in composition, those 2 points fit pretty close together. And then this desert community, because it looks different, I'm going to put on the other opposite side of the plot. And so we have analytical techniques that allow us to take thousands of samples and reduce all of that information about the microbial community into a plot. And if you color your samples, or you have different symbols here on the plot, then you can ask questions like, does your composition sort out by the type of habitat that you have been sampling from? So I'll give you an example. So here, my fabulous STAR graduate student, Kendra Walters, created this figure, which was looking at soil communities around the world. She used a very large data set called the Earth Microbiome data set. There's now over 26,000 samples where they have microbial sequence data online and available to analyze. And Kendra reanalyzed this data to try to ask the question, well, not just are soils different, but which soils in which type of plant habitats. And you can see, because the different symbols of different colors and shapes are generally clustering together, there is a pattern here. So communities from one type of soil, like in forest, are more similar to one another than communities such as in an agricultural field. Okay, well, you may have heard the word microbiome before, because you've heard about it in the news in terms of human microbiome. And indeed, since the DNA sequencing revolution has also then revolutionized microbiology in humans. And that's because since 2008, when the Human Microbiome Project got off the ground, so really only 10 years ago, we've discovered that there's also an incredible diversity of microbes on and in us. In fact, people estimate about 10,000 different bacterial species, and most of these live on our skin and mouth and gut, lungs, so of course parts of our body that are in contact with the environment for the most part. And one point I want to make about this diversity, to put it into some perspective, is that the diversity of genes that the microbes that we carry have, they have about 300 times more genes in those microbes than we have in our own human genome. So then if we think about how genetically similar you and I are to one another, we're much more similar in terms of our genomes than we are in terms of our genes and our microbes. And so this is an important thing to think about when you're thinking about the idea of something like precision medicine. All right, so again, just as we see patterns now of the distribution of microbial composition across different environments, we also see that there is interesting spatial distributions of microbes on our body. And so this is just, this is a publication that was looking at the composition of microbes across across human skin, and you can see in general the pie charts look more similar up near your head than down by your feet and your legs and so forth. So there are some fascinating studies showing that there are predictable community patterns. Okay, so I hope I've convinced you then that we are a microbial ecosystem and we live in a microbial ecosystem. And so traditionally these fields really haven't talked to one another. They've been studied completely separately, environmental microbiology and human microbiology. But those microbes, as Frank said, they don't distinguish between these things. And I asked Kendra to make another plot for today because I wanted to put humans and the environment on the same plot. And because some of those microbes are actually shared between the environment and us, you can see, for instance, here in orange, down here, that's all the soil plot. The soil samples that you saw before, now they look, you know, on this scale, they look much tighter together, so they're quite similar. But in comparison, you can see the human gut, it's over here on the left-hand side. And I think it's pretty interesting. And then the human mouth, that's quite distinct from our gut, which is a good news. And then in the middle, what I think is great is the skin, human skin. It's a mix between soil and human signature of those microbes. And the yellow, if you can see, if you're not colorblind, I'm sorry, Brandon Gott, then these air samples that are in yellow here, they're also right on top of all the skin. So really, we're in a mix of all these microbes, and they're sorting out into some different, there's definitely patterns in these different habitats, but it doesn't really make sense, I would say, to just partition these into one or the other fields. All right, so as an ecologist, you know, I started out in ecology studying plants and animals, and I was really fascinated by the idea, by the questions of how biodiversity, the variation in all these species, affected the functioning of an ecosystem, like in the tropics. How does the tropics work differently than a grassland here in Southern California, and how are those mix of species contributing to the way the ecosystem is working? Well, now maybe I've convinced you that essentially we have a tropical ecosystem in a handful of soil or in our gut, and we still want to apply those same questions. What is all that diversity doing? Is it affecting the functioning of the ecosystem and the health of that ecosystem? So in the environment, ecosystem functions, microbes play a role in the functioning of many processes that humans really depend on. So for instance, we care a lot about soil fertility, and microbes are processing nutrients, regenerating those nutrients so that we can grow new crops. They're decomposing waste, they're purifying our water, and so forth. But of course, we could just imagine that maybe it's just the sheer abundance of those microbes that make up that functioning, right? So a handful of soil, it has 1 billion cells in a handful of soil. And of course, if we removed all of the cells there from that handful, then of course, then the functioning of that soil would change. But the question is, does it matter which microbes are there? Because there are 10,000 species in that handful. And does it matter exactly which species are how abundant and so forth? And there's been a pervasive view in ecosystem ecology that generally composition doesn't matter, that whether we have one of these pie charts or another, it wouldn't really affect how the ecosystem works. And I think the general reasoning behind that is because diversity is so high. Because it's really hard to imagine that if you lost one species, that one of those other 9,999 species couldn't just jump in and play that similar role. So is it really true that just a small shift in a community composition would make a difference? So this is a lot about, this really gets at some of the questions that my lab tries to answer. And it's a convenient assumption to think about communities not mattering, because this is a really complex system. It would be really nice if we didn't have to think about all that complexity, if we could just predict what will happen to that soil if I warm it or dry it out. How fast will decomposition go? If I don't have to understand the community there, that would be a much easier question. But let's think a little bit harder about this question, and I'm going to introduce two different ecological terms here to explain how we're going to do this. And those terms are resistance and redundancy. So if I have a community, and then I apply some disturbance, let's say increased temperature, the question is, first, will that community change? Maybe it won't change. And in that case, we would say that this community is resistant to the environmental change. On the other hand, maybe it will change, and perhaps just slightly in terms of the relative abundance of the community. And then we would say this community is sensitive to change. But just because it's sensitive to change doesn't mean that its functioning will change. So we need to think about a separate graph. Here, and we often measure a function, not just is decomposition happening, but we want to know how fast is it happening. So we're measuring a functional rate. And so that's on the y-axis. Then we might also measure that function over time or across an environmental gradient, so we get some sort of curve. So if we're going to say that two different communities perform the same function, then essentially we're saying that they both have this same functional curve. If this is the case, we would say that these communities then are functionally redundant, that there's no difference in their function. Alternatively, maybe they have different functional curves, and so they might both increase their rate of this function, but they do it at a different rate or in a different shape. And in that case, if we change the environment, we wouldn't be able to predict based on one community what's going to happen. We'd have to know something about the second community as well. Okay, so we wanted to test some of these ideas, and one of the places we've been doing that is at Loma Ridge in the Loma Ridge Global Change Experiment. And this experiment was set up in 2007 by Mike Goulden. He's in the Earth System Science Department here at UCI. And it's a huge amount of work. And this is about half an hour away from campus. If you just drive up Jamboree and you kept on going and you got through this giant gate with about 10 locks on it, you would get to this part of the Irvine Ranch Reserve here. And Mike established this, and what you can see is one of the things he's manipulating up there is he's manipulating precipitation. And so what you're seeing is these shelters, and every time, actually not every time, about half of the rainstorms, when there's about to be a rainstorm, Mike in his lab will go up and put these giant covers over these metal structures to block the rainfall onto these plots, onto half of the plots. And so there we have then some plots that receive ambient rainfall and some plots that only receive about half the rainfall because they only receive about half of the storms that come through. So it's a ton of work. These are very large plots. This picture is taken from far away. And what he's been interested in is how the plant communities are shifting. You know, how might Southern California ecosystems look in the future as we expect more and more drought? So along with my collaborators here, we've been trying to study the microbes in this experiment. And so I really can't say enough about my amazing collaborators, Steve Allison, Adam Martiny, Kathleen Treseder. They're all amazing environmental microbiologists, and they all have their own expertise that contributes to these projects in incredible ways. And their labs too, and all of their students and postdocs have really contributed. So I'm gonna say we, but I really mean them, and all the students that have contributed to this work. Okay, so along with Steve, Adam, and Kathleen, we've been looking at one particular function, and that's decomposition. And we're looking at decomposition specifically of leaf litter. So these are just dead leaves. So you know, in your yard or across the UCI campus, you see all these leaves fall down, and then the gardeners have to come and blow them and remove them. Well, that doesn't actually happen out in these natural ecosystems. And if it wasn't for decomposition, all of that leaf litter would just build up and build up, right? But it doesn't. And so what's going on is this tremendous process every year of all these microbes degrading this plant waste. Now, In the grassland at Loma Ridge, it doesn't look like these big leaves. It's mostly grasses. And if you look on our hillsides here, you'll see a lot of that is standing up. It's dead plants, as well as some trampled down, but eventually it's being decomposed. Okay, and this is a really important fundamental process to the way our ecosystems work and even the global cycling of carbon. And so I won't get into too much detail, just to say, though, that as organic matter from photosynthesis and animals, when plants and animals die, that organic matter can either go into the soil or it can be decomposed by microbes. And when it's decomposed, microbes release the carbon back into the atmosphere because they respire CO2. So by studying the leaf litter, the surface layer of the soil, we're getting at one of these key points here of whether that carbon is going to go into the soil and be stored, or it's going to go back into the atmosphere. And of course, what we're interested in for our talk today is whether it matters what the microbial community looks like for that rate of decomposition. Okay, so there are a lot of different things that affect decomposition of leaf litter. Of course, the environment directly affects decomposition. As you might imagine, if there's less water decomposition and the rate of decomposition goes down, if you have a composter in your backyard, you would be able to see this. If it's too dry, things are decomposing much slower. The plant community also affects decomposition, because if you put different things in your composter, it will either speed up or slow down, because there's different chemistry of those leaves. And in fact, we know from a lot of Mike's excellent work at Loma Ridge that also the environment affects the plant community. So under drought conditions, the types of plant species that do better and grow better are different than those that are under ambient conditions. So these are all important feedbacks. But what we wanted to focus on then is how the microbial community fits into all of this. Because there's potentially, so there's those two questions. Is this community resistant and is it redundant? And so the first question, is it resistant or sensitive? If it's sensitive, then we might see that the environment, that drought, affects composition. And we might also see that the plant changing would affect microbial composition. And then secondly, if that composition changes, is it functionally redundant or does it affect decomposition? Okay, and if you're sitting there thinking, okay, this is all right, she has nice slides, but I mean, dead leaves decomposing, like how interesting can this possibly be? So I'm gonna give you a teaser, a reason to stay awake for the next 10 minutes or so, till we get to the exciting human microbiome side. But the teaser is that this exact same framework is what we're applying in humans. So just like we have environment and plants affecting microbes and functioning, we also have the environment or the host environment affecting the microbes, affecting your gut health, and you also have your diet. And so we're going to tease the same sorts of things apart in humans. But hold on to that thought, because we still have a little bit of leaf litter to go through. And so our first question, are the microbial communities sensitive to drought? So to answer this is pretty straightforward. Since Mike had done all the nice work imposing drought in these giant plots, all we had to do was go out to some of his plots, the ambient plots and drought plots. He has lots of replicates, takes leaf litter samples. We've been taking them every season for now over 7 years. And we characterize those samples. We characterize the bacteria and the fungi in those samples by DNA sequencing. Okay, to make a very long story very short, they are sensitive to drought. Here is a plot of the fungal communities and the drought communities. All those symbols that are in orange, those are ones taken from drought plots. And in blue, they're taken from non-drought plots, ambient plots. And you can see they're partitioning perfectly in this plot. So they're definitely sensitive, changing their composition under low water. But does it matter? So if I was a plant ecologist, and plant ecologists have been very interested in this topic for many years, I would, if I wanted to know how plant diversity, plant composition affected ecosystem functioning, this is how I would do it. So this is a very famous study in Minnesota, started by Dave Tillman now a couple decades ago. It's an aerial picture of a field that he has divided into these large plots, and he's planted different communities in each of those plots. So you can see that the plots are different even from the plane because they have different number of species and different community of those species. And so if you have this in a very nice replicated way here, then you can go out at the end of the season and measure different measurements of functioning. How is nitrogen cycling? What is the plant biomass? Other sorts of functions that you care about. Now, I'm not saying that this is simple. It's actually extremely difficult, and why it's such a famous experiment, because it's incredible that he's been able to do this for so long. In fact, it takes about 50 undergrads a summer, I hear, to go out and weed those plots to take out the weeds, because of course they're mixing all the time. But for microbiome research, it's even more of a challenge, right? But somehow we need to tease apart. What this experiment does beautifully is it teases apart the correlation between the environment and the plant community. Because different habitats have different plants, but they also have different environments. And exactly what we need to do is tease that apart here. But it's pretty hard to imagine an experiment like this in the field. How do we manipulate these microbes? If we put them out there, we can't see them. We're not going to be able to weed the ones that come into a plot, the same way you do with the plant. They're also extremely diverse. We're talking about, even if we could grow them, it's thousands of species. And also, I haven't heard of any microbe-free ecosystems, really, that we could do these experiments in, and certainly not microbe-free humans. So we're starting from a difficult spot. Now, the way we've approached this question is to use what we affectionately call microbial cages. So really, the first version of this type of experiment looked like this in my lab. And this is a cage made out of a dialysis tubing. And so it's a membrane that allows for water and nutrients to permeate through this membrane. And we could literally stick river mud inside the dialysis tubing and seal it up. And in this way, we could take different communities from different riverbeds, and then we could move them around and put them into different parts of the river. And so we could, in that way, tease apart the microbial community as a whole with the environment. And so the bottom picture are these different dialysis cages sitting in a riverbed. So my postdoc at the time, Heather Reed, pretty much almost killed me by the end of this experiment because it was really difficult and muddy and so forth. Luckily, Leaflitter is easier to work with, and so we went with these Leaflitter bags. And ecologists have used litter bags for a long time, but typically they're in window screen, and so you can put Leaflitter in them, you put them out into the environment, you wait a while, you come back and you weigh them and you say, how much leaf litter has been decomposed in this bag? So now we do the same thing, but we just make them out of membranes that are so small that bacteria and fungi can't move in and out. And so this is what we do. We take leaf litter from our different environments, we sterilize it, we autoclave it, gamma irradiate it every which way we can to remove the bacterial and fungal communities that are there. Then we put them into these, and then we put them into these sterile litter bags, and then we inoculate new communities on top of them. So it's a sterile leaf litter, and we're putting the community from wherever we want. We seal them up, and we place them back out into the field, into the environment we want. And in the study I'm going to tell you about, it's into different plot environments, an ambient plot versus a drought plot. And so all of this, then, it becomes a pretty complicated study, but it's essentially a three-way factorial study where we have different leaf litter, ambient and drought, different microbes, ambient and drought, and different plot environment, ambient and drought. And so if you cross all of those different which ways, you have 8 different treatments. We have many replicates of that treatment, and we can try to tease apart these 3 factors. Okay, so the first thing I'll show you then is that drought alters decomposition. And this is not surprising. As I said before, if you lower the water, decomposition rate should go down. So if we compare all of the leaf litter bags that were in the control plots, they lost just over 20% of their mass during this year. And then if you compare them to all of the bags that were in drought plots, they lost a bit less. So this is significant. By the end of the season, they kind of caught up with one another, but this was not unexpected. And then we compared, well, what about just the litter bags that had the drought microbes versus the drought versus the ambient microbes, regardless of which plot they were in. In fact, it had as big, if not bigger, effect on decomposition rate, depending on where we got those microbes from. So microbes from drought plots, they degrade the leaf litter more slowly. So we were pretty excited about this result because it suggests that microbial composition really had a surprisingly big effect, as big in this year as the actual environment did. So we saw then that leaf litter microbes are sensitive to drought, both through the environment, and I didn't show it, but also through the plant community, and that they aren't functionally redundant. They change functioning of the ecosystem. Okay, so this worked so well, we kind of went a bit crazy. We decided we were going to scale up, because if the microbes just in one plot versus another right over here in the same grassland, if that had such a big effect, what would happen if we took communities we know are much more different from one another? And so we thought, okay, where could we find that in Southern California? We decided to go along an elevation gradient, which varies a lot in climate, just here in the San Jacinto Mountains. So we have 5 sites all the way from desert up to the subalpine. And of course, these sites, they differ in a lot of things. They differ in the climate, temperature, and precipitation. They differ in the plant communities, and they differ in decomposition rates, so how fast that leaf litter is degrading. And of course, we want to tease all that apart with the microbiomes, because the microbiomes communities are definitely different. So this is work led by my postdoc currently, Sydney Glassman, who's about to take a faculty position at UC Riverside next month. And Sydney showed very clearly that the bacteria and the fungi are very different at these different sites. The closest communities are the pine, oak, and subalpine, but even those are distinct enough to recognize. Okay, so here's our crazy setup here. We decided we want to tease apart again the environment and the microbial communities. So we have these 5 different environments. And in each environment, we're going to put out litter bags, and we're going to put each of the 5 different communities in the litter bags on sterilized leaf litter. And just to make things somewhat tractable, we only use one kind of leaf litter. We use the grassland leaf litter here. But we had each community in each of these 5 different environments, and we wanted to know how important was that for decomposition. And here's just some pictures of those out in the field. You could see just based on these pictures how different those environments are. The desert is extremely low water, extremely hot, not much plant biomass around versus the subalpine site. Okay, so the results were very surprising, but also very interesting to us. I'm first just showing you the results here for the endpoints of the climate gradient. So the desert communities, and then the ones at the high elevation, the pine, oak, and subalpine. Because these extreme sites, they actually acted very similarly. So if you look, again, we're looking at mass loss on the y-axis, and these are the different sites. The desert, not surprisingly, decomposed the least after 1 year, and decomposition generally increased, maybe leveled off here at the high sites. But if you look at the 2 intermediate communities, then you saw something very different. In fact, the grassland communities did the best in the scrubland. In fact, they decomposed 40% more than any other community in that same environment. And alternatively, the scrubland did the best, scrubland microbes did the best in the grassland. They decomposed at least 20% better than the grassland microbes. So I'm not going to go much further. I could give you a whole nother talk on why this is, because that's the next question. If we show that microbial composition matters to functioning, we'd like to understand why that is. What are the traits of the microbes that are different, so that we could try to start making some predictions. And I will just tell you about one speculation we have, which is that they're probably differing in the types of genes that they carry for degrading carbohydrates. So leaf litter is mostly, and plants are mostly carbohydrates, and so they are made up of all sorts of crazy compounds. And microbes have genes that specialize on breaking down these various different types of chemical bonds in these compounds. And so that's where some of this work is going. We have students that are really focusing on the chemistry side and on isolating some of these microbes and testing whether they do in fact have different traits that can explain some of these results. All right, you made it through the dead leaves section. And so did I. I'll take a sip of water. All right, so much of what you've heard about microbiomes, though, is probably with humans. And so the idea is, again, thinking about composition, that the composition of human microbiomes is correlated with a lot of different health conditions. Everything from obesity, cancer, neurodegeneration, pretty much anything you can think of, vaccine efficacy, pharmaceutical use, so how effective a pharmaceutical is when you take it is somewhat, seems to be dependent on what is correlated with what microbiome you have, all of these things. But again, these are correlations. So we're kind of caught with the same problem here. How do we tease apart the effect of the environment versus the microbiome? Because, for instance, we see that there's a correlation between our gut microbes and obesity. But if it's just a correlation, it means a lot whether it's just a correlation or causative, because of course, if it's causative, then we might be able to actually manipulate the microbiome and improve health, right? So is it possible that we could actually change your microbiome to a lean microbiome, for instance, and it might help you lose weight? And the only way we're going to know that is if we start to tease these things apart. All right, so here's what, as I promised, the framework applied to humans. So we have the environment, which now, if we're thinking of the gut microbiome, is the host. So that includes the genetics, whether you're taking antibiotics, whether you're sick, anything about your condition, that can affect gut functioning. That's very well established. Also your diet, what you eat, plants or animals or a combination of both, that's going to affect your functioning of your gut. But again, the big question is whether those things affect your microbiome, its composition, and ultimately whether that change in composition is functionally important. So to demonstrate where people are on the human microbiome with all of this, I'm going to take you quickly through 3 different studies that I think kind of highlight where things are going in the field. The first is about colon cancer. And so this is a pretty widespread disease. It affects 150,000 people a year in the US. It's considered a westernized disease, and that's because it's associated with countries that have access to a lot of processed food. And so in the study I'm going to tell you about, they compared 2 populations that are somewhat genetically similar. African Americans here in the US and in Pennsylvania, where the rate of colon cancer is 65 out of 100,000 people, and rural South Africans where the rate is less than 5 per same number of people. And so what this study did was one of the things that obviously is quite different between these 2 populations is their diet. In the rural South Africans, they're eating a very low-fat, high-fiber diet. And as you might imagine, in the US, they're eating high-fat, low-fiber. In fact, I just like to put up these graphs because it's really astounding. So the South Africans were eating over 4 times as much fiber per day as the American population, and the Americans were eating over twice as much fat. And so what they did was, sorry, not yet. So let's talk about fiber. So the cool thing about fiber is it's carbohydrates. If you remember, Leaflitter, carbohydrates. And I like to tell my, my lab that, that digestion is just decomposition inside of you. So it's really the same thing. And fiber is made up of all these carbohydrates that we can't digest ourselves, that we require the microbes to digest it. So we don't have the ability to digest it. And so when microbes digest these, they produce these compounds called short-chain fatty acids, CFAs. They're the good, SCFAs. These are the good compounds, as far as we know. They're decreasing inflammation in your gut, and they're hindering tumor growth, or thought to, at least in some lab studies before this one. Then there's animal fats, which microbes do process as well. And when they process them, they release secondary bile acids, which are known to be carcinogenic. So there's good reason to think that diet and microbes might be involved in the incidence of colon cancer. Okay, so this study, what they did was they literally went to these places and they cooked the diets of the other people and fed them their meals. And so just for 2 weeks, the South Africans ate the American diet and the Americans ate the rural South African diet. And what they saw was pretty incredible. They saw that microbial composition changed in that 2 weeks towards the fiber degraders of the Americans that ate the fiber diet. Inflammation went down, and even markers of cancer risk went down in just 2 weeks. And the exact opposite happened in the poor South Africans, who probably after 2 weeks were done with that diet. All right, so this experiment is one of many experiments now that are showing directly that diet, of course, affects gut functioning, but also that diet is affecting the microbiome. But I would argue that it's still not making this final link between the microbiome and gut functioning, because we still can't tell, is it that the microbes themselves are the ones that are altering the cancer risk markers, or could it be a direct effect of the diet? All right, so this second study gets a little bit closer, and this study looks at people with type 2 diabetes, which, as many of you probably know, this is the inability to use insulin properly, and insulin is very important for regulating the sugar in our cells. This is a huge problem in the US and only growing. About 30 million adults have it, and in the last 20 years or so, you can just see the trends here on these maps. So all of those deep red states in 2015, they had over 9% of adults are diagnosed with type 2 diabetes. And so this Rutgers researcher, he was looking just at patients with type 2 diabetes, and he had a control population, and then he had a population that he fed a high fiber diet. He did this for 12 weeks, and it changed their microbiome, as we would expect, increased those good compounds, and decreased blood glucose levels. But then he took one step further, and he transplanted these microbes then into genetically similar mice. So they're the same host, genetically identical, but now they have the human microbiome, and fed them the same diet too. So now there's not even a correlation with diet. And he found that this improved the blood sugar of the mice. So this is, again, one step further, but we're still talking about mice. So let's think, you can see where this might be going. It's getting messy. But the last study I want to tell you about is Clostridium difficile infections. And many of you probably have heard of this really problematic pathogens, also called C. diff. It's ubiquitous, so most of us have it and carry it, but especially when you take antibiotics, it can overgrow in your colon and take over the diversity there. And it's quite common in hospitals and nursing homes, and it causes all sorts of problems, and it can even lead to death. So in a few dire cases some years ago, doctors decided that they were going to try, last resort, try a fecal transplant from a healthy human donor. And so you have a C. diff infection, your microbiome is really reduced in diversity, overgrown by C. diff, and then transplanting from a healthy donor a fecal transplant. Across the globe now, just summarizing all of the experiments, there's been about a 90% cure rate. In fact, they've stopped a lot of these studies because it's actually unethical to not give the other patients the option for the transplant, as opposed to traditional treatments of just antibiotics. So this has been quite astounding. Of course, it's quite messy procedure here, so we would like to improve upon this. But I will say that this is ongoing now here, even at UCI. So Dr. Nimisha Parekh is a gastroenterologist at our medical center, and she's been performing this for patients. But to try to improve these things, one thing is that it's really hard to find a healthy donor really quickly, because you do need to screen your healthy donor. You don't want to be introducing new pathogens to a very sick patient. And so of the projects ongoing now is my colleague Katrina Whiteson, also in the School of Biological Sciences. She's working with Dr. Parekh to study the use of OpenBiome. And OpenBiome is a company that pre-screens healthy donors, donating their poop to this company. And so that if the doctor decides you need a transplant, you can get that right away, and you don't have to wait the week or two to screen a healthy donor. And so they're working on trying to understand, what is it about this transplant? Why is it working? Because it would be nice to grow the important microbes here and put the cultures together into a pill, right? And basically separate it from all the other crap. Okay, so this leads then to the interesting question of whether we can engineer the human microbiome. If we now see that it's not just a correlation, but it really is causative, then this just opens up the idea of trying to use microbes as promoters of health and combaters of disease, and can we engineer that community? So I hope even in these short little vignettes, I've convinced you that one way you can go home and start engineering your microbiome is by changing your diet. And of course, I would recommend eating fiber. Katrina and I have a mantra now that's lentils and beans, pretty much, if you ask. That's what you should be eating. You can also try probiotics. So this is where you take cultures of microorganisms from a lab, put them in a pill, or in yogurt, for instance. Lots of these are on the market now. And many claims have been made about them and how they might improve your health. I will note, however, that almost all of these, none of them are actually gut microbes, right? So they're not going to recolonize your gut. A dairy microbe, not to say that it's not having a health effect and improving your health in some way, but it's not colonizing your community and shifting that. So there's a lot of work to be done here in this area, and whether we can identify bugs that actually can colonize in a good way and improve our health that way. Of course, as I was just mentioning, perhaps I think what would be really interesting is trying to develop cocktails of these different microbes that could colonize your gut and improve things that way. Okay, so this leads me to think about, what are the big questions here, and I really want to stress that I think they're very similar for both humans and in the environment. These questions like, what is a healthy microbiome? Not just what is correlated with health, but what is even a microbiome that might be resistant to getting disease in the future? We really have no clear idea, both for soil and for the human body. Can we promote the development of healthy microbiomes? So here I'm thinking a lot about children. What should we be doing to promote the development of an adult healthy biome already in our kids? And finally, if it goes wrong, what can we do to alter it, as a last resort and bring people back to a better steady state? So I'm just going to end telling you a little bit about the UCI Microbiome Initiative. And as Frank said, it really is drawing upon Katrina and my experiences trying to bring together these two different fields of human microbiome and environmental microbiome research. And it just started last July, but already there are 55 different faculty involved somehow, and they come from 9 different schools across the university. So we've just been amazed and a little overwhelmed at the interest in this, but it's been really exciting. And so if you think, so we just think there's a ton of opportunity here to think about microbiology or microbiome science across systems. And that UCI has this opportunity because we have this amazing group of environmental microbiologists already here, and we have strengths in all these different areas across campus, and where they connect could really promote a lot of excitement and synergies here. So we have a variety of goals, including research and strengthening research on campus in this area, training microbiologists. And I'll just mention a couple here. So if you're a faculty member or grad student postdoc and you're interested in including microbiome science in your project. We have lots of resources up on our website. We host a consulting hour every week. That's pretty popular now, so we now have a sign-up sheet, so not 5 people don't come at the same time. But we host monthly meetings. We've had pilot projects we've been funding and so forth. So look out for those and sign up for our email list if you're interested. We've also been hosting workshops and teaching people hands-on how to do some of the techniques so that they can do this with their own samples, how to analyze the data. These are just a couple we've had this year. And on the training side, we've had some fun experiences here. You know, I think, again, there's a huge opportunity where we could be a leader at UCI and in moving microbiology in this way, coming at microbiology from an ecological perspective and thinking about it in a community perspective. And so one of the ways we've been doing that is working with Julia Massamelli, who's a faculty member in the school, along with Alex Chase and Andrew Oliver, who are graduate students, and Katrina in my lab. And we had her class, they had to volunteer to be in this lab class, and then they agreed to undergo a 2-week diet intervention. So what do you think the diet intervention was? Lentils and beans, yeah, pretty much. Actually, if you're an undergrad, we had very fascinating discussions with the graduate students about how we could get undergrads to eat more fiber. You know, we wanted them to eat 50 grams of fiber a day. How could we do this? And actually, it's getting late, so you should probably ask me at the reception some of the different types of things we considered. But in the end, we chose to go with a meal delivery service. And we partnered with Thistle, and Thistle provided a very discounted rate, 10 meals a week for each of the students. And they actually are the only delivery service I know that actually does have 50 grams of fiber a day in their meals. And so in that way, not all of them got to 50 grams, but they at least definitely increased it. Especially wasn't hard when many of them, the week before, where they were collecting samples and they had to put in MyFitnessPal what they were eating, some of them had 0 grams of fiber a day. It was like, God, you have to avoid it. It's just literally pizza and beer or something. So I don't have the results it was just this quarter. It was super fun. These guys did a ton of work. I really had nothing to do with it, but we're really excited because I think it's going to end up being a publishable paper showing how the fiber diet changed their microbiomes. Anyway, the students really, I think, got a lot out of this class. So I just want to acknowledge then, again, my amazing collaborators, and both here at UCI and other folks around. And then on the right-hand side, most importantly, my recent grad students, postdocs, undergrads that really make coming to work fun every day. Thank you so much. This is all their work too. And thank you all for listening.